In the last, I should say, in the last 20 years, there was plenty of information referring that the importance of conformational diversity in a huge amount of biological processes related with protein activity. During, for example, enzymes, catalysis, signaling, talking about specificity, promiscuity. These are the typical examples of the existence of conformational diversity, cooperativism and allosterism, origin of new functions during evolution, protein-protein interactions, etc., etc., etc. And a huge list of importance with examples bolting the importance of conformational diversity in protein biology. So, about 10 years ago, more or less, we started to generate questions about the relationship between conformational diversity and different aspects of proteins. The very first, I'm a biochemist, more or less I'm a bioinformatician, and a structural bioinformatician. And at the very beginning, the first idea was, okay, we have to have a large amount of proteins with different degrees of conformational diversity. That was the first point in our research plan. So we have to obtain a large data set of proteins with experimentally based conformational diversity. And then we started to ask different questions. Which is the distribution of conformational diversity in the known protein space? So proteins are very dynamically, they behave very dynamically, they are rigid. That was one of the questions. And different questions is there any relationship between biological function and the extension of conformational diversity? Maybe a protein can move like this, and others can move like this. So is there any relationship with the function of the proteins? How conformational diversity depends on sequence variation. Or, for example, which is the number of conformers and its distribution. I told you that hemoglobin has 2 main conformers, but maybe other proteins have 3, 4, 5 different conformers. And in the end, can we predict, as a good bioinformatician, can we predict conformational diversity using sequence information? I'll tell you. So, the first step, more or less 10 years ago, we create the first database of proteins having different proteins with different degrees of conformational diversity, and we call it CODNAS. Previously, in 2011, we designed another database, but this is more nice, in 2013. CODNAS came from database of conformational diversity and native state in proteins. And the main hypothesis we followed at that time is that, you know that protein structures are in a very well-known database that is called PDB. PDB is a huge database with all the proteins with known crystallographic structure, or NMR structure. So we took PDB and we collect a redundant collection of structures for a given protein. Redundant collection. So here maybe you can find 4 different structures, 4 different crystallographic structures for a given protein with 100% sequence identity. So maybe this structure was crystallized with the ligand in the apoform, another in the oloform, another with an inhibitor, another with an allosteric inhibitor, or whatever, a different pH. Okay? As the conformers are in equilibrium, the existence of a ligand shifts the equilibrium towards a given conformer. Okay? So we can take all these structures that may differ in each other as a snapshot of protein dynamism. Okay? This was also checked using NMR and crystal structure. Okay? So these are instances, snapshots, yes?
So how do you explain that those are artifacts of the crystallization process, which is a really, I mean, because you have to dehydrate the protein. So this is something that I always puzzle.
The main problem with this, dealing with the structures and conformational diversity, or as useful input in considering the dynamism of this, is crystal contacts, for example. That maybe in the same crystal you can have another molecule that interacts with one of the structure. But we did a lot of correlations, and we found no correlation between the contacts as an artifacts in the crystals with the conformational diversity of the protein. Besides that, we have no other possibly check. I mean, here in Coordinates we have about 25,000 different proteins with 20 different conformers in average per protein. So maybe you have chances, of course, to have different errors, of course, but I'm a bioinformatician, and so I use a huge amount of information just to do the... A little bit of dynamics is from the free energy shift, so can be gathered. So have you never tried to see conformers? Yes, there are plenty of papers that, for example, doing molecular dynamics, doing NMR, that proves that, okay, these different structures correspond to the same ensemble. That, of course, it was checked. Yeah. So here I superimposed all these different conformers that was obtained in different conditions. Okay. So then we collect all this redundant collection of different structures for the same protein, and we estimate, for example, to measure the structural differences between the conformers, the RMSD. The RMSD is simply the root mean square deviation of the distances between carbon, alpha carbon, in the backbones of the proteins. Besides that, we estimate also additionally different parameters as TM score or GDT. GDT are different parameters to measure the structural similarity or structural differences between different structures. In these cases, different conformers. But also, we collect information about the crystallization conditions. So, for example, pH, temperature, presence of ligands, presence of mutations, the oligomeric state of the protein, post-translational, presence of post-translational modification in one of the conformers, and so on. So we can relate the extension of conformational diversity measured, for example, with the RMSD, with given parameters of crystallization. For example, I can have I don't know, 1 or 2 angstroms of RMSD due to the presence of substrate. Suppose if I am comparing the holoform and the apoform of a given protein. Okay? Of course, we gather different information from other databases as structural classification, information of different sort, taxonomy and whatever, function, characterization of enzyme reaction, etc. This is almost a little updated. More or less, we have about 25,000 different proteins in coordinates nowadays. This was about performed 16 million different structural alignments between all the conformers, are more or less now it's about 17 point something, the number of different conformers in average that we have per protein. So this is the interface of coordinates. You can have here, you can display the different conformation of a given protein, and it's additional information there. And also you can study the profile of the main differences as a function of the position between two given conformers. We also display the sequence. You can see here that the sequences are identical. So these different structures represent different conformers. So it's important to know that when we structurally align all the conformers for a given protein, we record the maximum RMSD. And that RMSD, that maximum RMSD, we take it as the indicator of the conformational diversity of that protein. Okay? The maximum RMSD.